sciences. She has expertise in cardiometabolic health, gut microbiome, and omics techniques, in clinical nutrition interventions, and pre-ingestive controls of nutrition. Her work has contributed to the beneficial effects of functional foods such as almonds on vascular adiposity and cognitive function in adults with overweight and obesity. More recently, her work has characterized benefits of chronic almond consumption on blood glucose regulation, gut microbiome, and metabolism, assessed via metabolomics and transcriptomics techniques. Her current research program is focused on using systems biology approaches to understand the effects of dietary patterns and exercise on human health and host microbiome interactions, and in alleviating health disparity. She has grants from NIH, the Almond Board of California, and MU Genomics Technology Core. Before I turn over to Jaapna, I want to remind the audience that if you have questions, you can post a question in the Q&A session. So right now I'm going to turn over to Jaapna. It's all yours.
Hey, thank you very much. So and thank you for the very wonderful introduction, Mingxi. I'm really excited to talk about my research portfolio today. And, I'll be talking about several studies that I'm doing here at Mizzou, and then some of the studies that I've done in the past, and then of course, some upcoming projects. So just a little bit about myself. I got my undergraduate degree in bioinformatics actually from India. And that was a while ago, as you can see over here. And, it was around the time that I really got interested in nutrition and fitness. So I decided to get certified as a personal trainer. And, a lot of the nutrition knowledge at the time came from the internet, which I don't recommend at all. And luckily, I decided to pursue higher education in nutrition science. So I ended up going to Syracuse University to get my master's, and the focus of that research was more community nutrition and dietetics. But I was really interested in clinical work and specifically weight management. So I ended up at Purdue, where I got my PhD and specialized in clinical work, ingestive behavior, and biostatistics. After that, I did my postdoc at UC Merced, and where I really delved into the gut microbiome and using metabolomics in the context of clinical interventions. After that, this is during the pandemic. In fact, right when the pandemic had started, I transitioned to the University of Missouri-Columbia here, and I started my faculty position in July of 2020. And I'm really happy to be here. So my research overall, I'm focused on really personalizing lifestyles, for example, diet and exercise for optimal human health. And we humans, we don't exist by ourselves. We're an integrated system with the microbes that reside within us and on us. And so really, my focus of my research is looking at how lifestyle impacts us as an integrated system and impacts cardiometabolic outcomes, which I'll talk about more in a little bit. And that's the biological phenotype, but also what happens behind the scenes in terms of changes at the biochemical level and the DNA and the RNA levels. And then, another part of my research, which I won't go into too much today, is looking at how key environmental and pre-ingestive factors can influence food intake in general. So this is a really nice graphic that I found, which summarizes, what cardiometabolic disease indicators are, and really gives a good overview of what I am doing currently. So my work focuses on, looking at glucoregulation, how the body responds to insulin, specifically how sensitive your cells are to insulin, which is a hormone that's secreted from the pancreas. So insulin resistance and glucoregulation. and then blood pressure. So we've all heard of hypertension, and then abdominal adiposity. So, body fat composition, and specifically around our abdominal area, because that is the one that's more detrimental to health. And then, looking at blood lipids, so things like cholesterol in the blood and triglycerides in the blood. And a lot of these factors are affected not just by, dietary variables and exercise variables, but a lot of these factors are affected by other biological, behavioral, and environmental variables as well. So one aspect of my research, yes, is cardiometabolic health, but then I also work with the gut microbiome, and I'll give a brief overview of that before we actually delve into the study, so it's easier to understand specific terms that I use. So we typically collect stool samples to look at the gut microbiome. So stool is a good proxy for what goes on in the colon. And so once we collected the stool sample, the DNA is isolated from the sample, and it's the 16S RNA gene, which is what I typically do, 16S RNA sequencing, is amplified. And once these genes are amplified, we can use some bioinformatics to process the data, do some quality control, and really analyze the sequences that have been generated. So once we have that data, we're able to match it to online databases that have already identified those microbes. And we can come up with a taxonomic classification for the microbes. And then we can do other more advanced techniques, such as diversity analyses, and to see changes in overall microbiome patterns. And so I talked about this briefly, but we look at the biological phenotypes. We think of things like blood glucose and blood pressure, cardiovascular health. All that comes at this level. But I'm also interested in seeing what drives changes in those factors and what techniques we can use to do that. And so I commonly do use metabolomics, and which is essentially systematic study of the unique biochemical fingerprints that specific cellular processes in the body leave behind. So, and there are two ways you can do: targeted, where you look at specific metabolites in any tissue. So I study blood and adipose tissue, fat tissue. Or you can do untargeted, which means that you're profiling basically anything that you can find in a specific tissue. So I use both. And then I also do transcriptomics, where we isolate RNA from blood samples, and then we look at changes in gene expression profiles in blood samples following a specific intervention. But if you do it at the protein level, it's called proteomics, and at the DNA, it's genomics. So I'll talk a little bit about this post study that I did at University of California, Merced, where we looked at the effects of almonds on cardiometabolic health and the gut microbiome. And then I was also funded by the NIH to do some additional metabolomic analyses on this project as well. Now, these are just, it's a good graphic of how potential mechanisms of which almonds can improve metabolic health. And you'll see here that nuts are really, really intriguing, and almonds specifically have a good source and range of these beneficial nutrients that can have all these potential health effects that are interrelated. So potentially can improve glucoregulation, can improve your lipid profile, which in turn can improve, vascular health, can improve blood pressure. And you'll see this with dietary interventions as well, where you see improvement in one factor and then with a specific intervention, but you also see improvements in other factors, health factors, because they're all interrelated. And with recent advent in microbiome technology, and now several observational studies have shown that the microbiome is actually associated with each of these factors as well. So in this specific study, we recruited college freshmen. And it's a very interesting age group to study because they're nutritionally independent. They transition from this environment where they're dependent on their parents or the caregivers into an environment where they can pretty much do whatever they want. And you've probably heard of the freshman 15 phenomenon, which states that freshmen gain on average 15 pounds their first year. That's a little exaggerated. It's not 15 pounds, but there is considerable weight gain. And along with weight gain comes increased risk of metabolic disease as well. And there are alterations in eating behavior. So that's why we chose this specific population to study. And one of the advantages of that is that they were just 18 to 19 years old, and so very homogeneous population in terms of age as well as their physiology. And plus, because they were mostly on campus, we could closely monitor their food intake and track compliance. So these are just the baseline characteristics of the participants who were enrolled in the study. So before the study, what they looked like, and as I said, pretty homogeneous. And UC Merced is categorized as a Hispanic Serving Institute. So most of the participants in the study were Hispanics, and we had limited enrollment from other racial and ethnic groups. And I think this study was a turning point for me in terms of really looking at the representation of different racial and ethnic minorities in clinical interventions, because they're typically underrepresented. And so, when I was working on this study, that's where I decided to really get more into health disparities in these groups as well. So in this specific study, this was single-blinded. So, we participants, well, we didn't know which group they were in, but the participants did. And it was randomized. And so participants were randomized into either the Almond group or the control group. And it was parallel arm. That means we just had two groups. So it was an 8-week study. And so participants were randomized into the Almond group, or asked to consume 2 ounces of almonds while they're in the study. So 2 ounces is roughly 2 fistfuls, 2 small fistfuls. Whereas those that were randomized to the control group were asked to consume the same caloric amount of graham crackers, which is roughly 5 sheets of that, as well as refrain from consuming any nuts or nut products. So they were asked to maintain consistent dietary patterns, consistent physical use, and supplement use, because we want to ensure that nothing else is confounding the results. So that study particularly, we saw the changes in fasting glucose in both groups. So consumption of almonds and graham crackers decreased fasting glucose in both the groups. No significant difference between the group. Specifically in terms of insulin sensitivity, that is how well your cells respond to insulin. So the main role of insulin is to regulate the amount of nutrients that are circulating in the bloodstream. So we actually saw improvements in insulin sensitivity with almond consumption, but not with graham cracker consumption. In terms of blood cholesterol, we saw similar decreases in blood cholesterol, total cholesterol with both groups. But almond consumption prevented the decline in HDL, which is typically the good cholesterol, but that continued to decline with the cracker group. And endothelial function, which is just a measure of how healthy your arteries are, we didn't see any changes with respect to almond or the cracker group. Now, looking at the microbial profile, so we looked at alpha diversity. Now, greater microbial diversity in the gut is more beneficial for human health. And this is Shannon's index is just a measure of one measure of alpha diversity. And here you see at baseline, there really wasn't any difference between the almond and the cracker groups, but 8 weeks after the intervention, the microbial diversity significantly increased with almond consumption, whereas it did not change with cracker consumption. So we were really excited to see these results. Next, we looked at changes in specific bacterial taxa or specific microbes over time. And the one that was most interesting was Bacteroides fragilis. So this is it's typically a commensal bacteria, which is that it doesn't really have a detrimental or a beneficial effect on human health. But what can happen is that if it's displaced into the bloodstream or the surrounding tissue, following disease or trauma, it can be pathogen. So what we saw was that, again, 8 weeks after almond consumption, the abundance of Bacteroides fragilis significantly decreased, whereas that of in the almond group, but not in the cracker group. So another interesting point. What we're doing right now is, and I don't have the data right now, but we're doing metabolomics to see, changes in metabolites that are involved in carbohydrate and lipid metabolism in response to almond consumption. And we just submitted that paper for publication. So moving on from, just a single food approach, the study that I'm conducting over here is really looking at how improvements in dietary pattern can impact glucoregulation. So, not single food, but because realistically we consume specific dietary patterns, we're not just eating one food at a time. So now my research direction is moving more in that direction. So in this study, what we're looking is how a personalized Mediterranean style diet can impact all these factors. And if you personalize the diet to an individual's, habits and some blood work that you collect based on, and the BMI, body mass index, weight, it can improve diet quality more than a non-personalized diet. And here, a Mediterranean style diet is a diet that, which is rich in fruits, vegetables, and nuts, and unsaturated fats and fiber. So we're trying to see if that diet can improve diet quality more than conventional dietary guidelines. So if you go to the MyPlate website, you'll see conventional guidelines listed. So if that can improve diet quality more, would there be a greater impact on cardiometabolic outcomes? Similar outcomes that I talked about previously, glucose regulation, insulin sensitivity, blood pressure, and if it can improve carbohydrate and lipid metabolism using metabolomics, and then gut microbiome as well. So that's one goal. And the second is to really see if improvements in these outcomes are differentially influenced by race or ethnicity. Now, there's an increased incidence of diet-related cardiometabolic disorders in ethnic and racial minority groups, specifically Black adults. And these groups are typically underrepresented in clinical interventions all across. And so, one of my research goals is actually to improve their representation in the studies that we do so that we accurately able to really look at changes by race and ethnicity. And there is evidence that, Black adults have greater insulin resistance and indices of hypertension, etc. So in this study, we have 4 diet and race groups. So participants are randomized to the personalized Mediterranean style intervention, and they get fruits, vegetables, and nuts to eat every day for 8 weeks. And then they're also counseled by a dietitian every week for the first 4 weeks, and then every 2 weeks after that. Whereas those who are in the conventional dietary advice group, they're just provided generic dietary counseling at the beginning based on the MyPlate guidelines, and they're not given any foods. So another thing that I want to point out with this design is that, if improvements in diet quality improve health outcomes in both Black and white people equally, but we find race effects in the control group, then that means there are other environmental factors such as socioeconomic status, food environment that could be contributing to these effects as well. And so that data we're collecting as well, separate questionnaires that we do to really capture the environmental aspect of these differences. The design looks somewhat like this. So we collect their demographic, anthropometrics, we collect fasting blood samples, blood pressure, do a lot of questionnaires at the screening visit, just so that we give that information to the dietitian who can personalize their diet. Then these participants undergo a couple of weeks of study training, and they randomize either into the intervention group or the control group. And then we collect a lot of these other factors. So we test anthropometrics every 2 weeks. That means weight and their body composition, fat mass, lean mass, things like that. And then we also do an oral glucose tolerance test, which is what we did in the Almond study as well, where participants are asked to drink 75 grams of glucose. It's a glucose drink. And then we take their blood before they have the glucose drink, and then 15, 30, 60, and 120 minutes after that. And so we'll be conducting, we look at changes in glucose and insulin over that time period, and then also changes in metabolomics profile over that time period as well. And then we collect stool for the gut microbiome and perform other cardiovascular measurements. So this is data from a very, very small subset of participants. So we collected this preliminary data. I think this is from last year, just 3 to 5 individuals per group. So we haven't completed the study. It's going to take another 1.5 years to actually complete clinical trial recruitment. So we see some interesting trends over here. So let me explain this. A Matsuda index is actually an index of insulin sensitivity. And so typically you want this to be higher, and you want the insulin resistance index to be lower. So what we see over here is that in Black adults, we saw an improvement in insulin sensitivity and a reduction in insulin resistance index in response to the Mediterranean style diet. but and these changes were actually more pronounced than that what we observed in the white group. So we're seeing that this Mediterranean style diet impacts glucoregulation differentially by race and ethnicity. Next, we looked at changes in microbial taxa, but before that, this is again a subset of participants at baseline, so before they started the intervention. And this graph over here shows microbial taxa. So these are just specific microbes, and these are just general functions of those microbes. So we want to know not just what microbes change, but also how the functions of those microbes change. And here we see that you see distinct race differences in some of these specific taxa and their functions. Now, in response to the Mediterranean style diet, we observed differential changes by race in the gut microbiome. And this is again another subset of the participants that have completed the intervention. So here, the red dots refer to the black adults. We just had 3 completed the study at the time, and the blue are the white adults. And before the intervention, we really didn't see any difference, but even as early as 4 weeks into the intervention, you see these clusters forming. And so then we decided to really go into the different taxa and see what was happening with the taxa, microbial taxa here. Now, we see differences again in specific microbial taxa in response to the Mediterranean style diet. The 2 that I want to point out over here are the Lactospiraceae, which is a family of short-chain fatty acid producers. And short-chain fatty acids are byproducts of microbial metabolism of dietary fiber, and those are important and beneficial for human health. So we're seeing that these producers actually, these increase, this family of producers increase in black adults, but then decrease. But then we didn't see much of a trend with white adults, maybe a slight increase over there. And then Doria formicigenerans is a specific microbe that's found in this family. And it's also from the family of short-chain fatty acid producers. And we see an increase in black adults, but not in white. So we're seeing changes happening over here, but what that means right now, we're not at that point as yet. So the next step is to complete recruitment. And then once we have all our data, we're going to perform mediation analysis to actually relate the gut microbiome with changes in glucoregulation and see if that is a mediating factor for the improvements in glucoregulation that we see in this population. So the next study that we're conducting, it's still going on. We're looking at how resistance training can impact the gut microbiome as well as glucoregulation. And exercise is essential in improving glucoregulation in obesity. And a lot of observational studies recognize that the gut microbiome is an important factor that can influence the variability in glucoregulation in response to exercise. And exercise and gut microbiome, gut health have been studied, but most studies have been on aerobic interventions and not resistance training, which is strength training per se. So in this study, what we're looking at is how resistance training for 6 weeks can improve diversity of the microbiome, increase production of short-chain fatty acids, and then also upregulate the metabolic pathways that those microbes are involved in. And whether, these changes that we see with exercise, improvements in glucoregulation, if those are mediated by the gut microbiome in these adults. So the adults that we recruited for the study are sedentary, so don't exercise on a regular basis. They're young adults and BMI of overweight and obese. And so these participants are randomized either into the resistance training groups. If in that group, they undergo a moderate intensity protocol, so the resistance train about 3 days a week for 6 weeks, whereas those who are in the control group are just following general physical activity guidelines, which we give them at the beginning of the study. And so my master's student, John Cullen, is doing an amazing job training these participants, and he's really been instrumental in collecting the data for this study. So I'll show you some preliminary data after the study design. I think by the end of the summer, we would have finished recruitment of all participants, and then we can start really looking at the data as a whole. But we collect similar outcomes at the beginning for screening, just to see if we can personalize the intervention a little bit more to the participant. And then we collect stool samples to look at the gut microbiome. We again conducting an oral glucose tolerance test, and then stool collection also for looking at the short chain fatty acids that I just talked about. And then we look at other measures of adiposity, body composition, and then cardiovascular health, appetite, etc. So just these are data just from 3 participants at the time when we had, and this is the glucose area under the curve over 120 minutes. And what we're seeing with this intervention is it's very important, like in any exercise intervention, to control for weight stability. So we're seeing here, like there's some participants who perhaps changed their diet, ended up gaining a little bit of weight, and in those participants we didn't see improvements in blood glucose. But in the participant that remained weight stable, blood glucose decreased. So, it's important to control for weight stability. That's what we're learning right now. And in terms of the bacterial taxa, I would like to talk more about Roseburia species. This belongs to a group of dominant short chain fatty acid producers again. And so we're seeing an increase in those bacteria that are involved in short chain fatty acid production with resistance training. So that's a good thing, and we're excited about that to see what the rest of the data looks like. And if we specifically look at not just the taxa, but the functions of those microbes, what kind of pathways that they're involved in, we see several changes in several metabolic pathways that those microbes are involved in as well. And one that is not presented on this slide is actually bacterial chemotaxis, and so that's a motion of bacteria towards a more favorable environmental condition, and we see an improvement in that with resistance training. And then we also see decreases in lipoic acid metabolism, phosphotransfer system, pyruvate metabolism with resistance training. So collectively, what this tells me is that it just suggests a more efficient carbohydrate and energy metabolism with resistance training. And so we're at the point where we just need a couple more months to finish data collection for this, but my future studies, we just submitted the proposal for that to NIH, is looking at the complementary and potentially synergistic effects of diet and exercise collectively on the microbiome. And so that intervention would be a joint diet-free Mediterranean style intervention, as long as a combined aerobic and resistance training intervention. So let's see how that turns out. And then I have a couple of upcoming projects. So we just got funded by the Almond Board of California to look at the effects of almonds on immune health and responsiveness in adults with obesity. And I presented some of the almond data from my postdoc work. And at that time, in a subset of participants, we did transcriptomics on the blood samples to see how almond consumption can change gene expression profiles. And we did some pathway analysis, and this is just a pathway map of the coronavirus, COVID-19. And so it was interesting that pathway never existed until a year or so ago, when obviously, because we've made so much progress with COVID-19 now that now databases incorporate that pathway as well. So what this shows is differential changes in specific gene expressions in response to almond and controlled. And there's some over here that inflammatory cytokines, inflammatory markers that are involved in immune health, that we see changes with in response to almond consumption. So this got us to think of, well, what else is there? What else can we do more targeted analysis? So we used this preliminary data to apply for a grant to the Almond Board. And I collaborated with Dr. Vicky Vieira-Potter. She's a co-I on this project. And what we're exploring is how almond consumption could improve blood and adipose tissue immune health markers, how it could improve immune responsiveness to viral infections. Now, it's not ethical to really directly expose humans to any sort of virus. So what we're going to do in this is we will extract PBMCs from participants' blood, both before and after the intervention. PBMCs are peripheral blood mononuclear cells. And so there are a variety of specialized immune cells that work together to protect our body from harmful pathogens. Now, in a laboratory stimulation model, we will expose those PBMCs to the influenza virus and see if that changes the response of these immune markers. So and if that is different before and at the end of the intervention, so after almond consumption. And then we'll also do some transcriptomics to see gene expression of immune markers. So we just got funded for this. We're working on the IRB application, and hopefully we get to submit that next week. And so this project will probably start sometime in the summer. And the next study that I would like to talk to you about is this study that was, it's not really funded by a grant, but we got some money from donors, the Slatons and the Rooney Times, to give us some support to look at the project, look at effect of different beef products on cardiometabolic health and the microbiome. And so this study is done in 2 phases. The first part is actually the animal study, and Dr. Jordan Thomas is the co-PI on this with me. He is working on the animal study right now. And so we're looking at 3 endpoints for finishing beef products. So they're being raised right now, the beef products. So we'll probably, for our human study, we'll probably get that product by the end of this year or early next year. So the first product is a conventional grain finished product. So these would be fully finished animals and probably have greater intermuscular fat. And then the second one would be forage finished product from the animals as well, around 26 to 28 months of age. And this is not fully finished, and it'll have lesser intermuscular fat. And usually how favorable it is, the flavor profile depends on the consumer. And then we'll have a fully finished product as well from animals with 26 to, I think this is supposed to be 20 to 22, sorry, and this is 26 to 28 months of age. So once we get these products, we will run a clinical trial where participants will consume either the conventional product, forage finished, or as a control, we'll also have a plant-based product. And then we are going to do some similar testing. So we'll do postprandial testing where participants will consume this, and then we'll collect their blood samples before they consume and after to look at changes in metabolomics profiles, look at changes in glucoregulatory profiles and fatty acid profiles. And so here, these will have different fatty acid profiles. That's based on the feed that these animals have been consuming. And we're hypothesizing that those fatty acids and lipids are going to manifest in the blood samples as well, right after consumption. But we're also interested in seeing not just postprandial consumption, but how these changes look like, or what these look like after a short 4-week intervention. So participants will eat this every day for 4 weeks. And we have a metabolic kitchen here, so our chef is going to prepare those meals for us. And then we'll again collect these blood and stool samples before they start the intervention, and then 4 weeks after. And we will track changes in, again, different metabolites in the blood, their glucose, insulin levels, lipids, fatty acids. And we also look at cardiovascular health and the gut microbiome. And based on what we get over here, we might submit a bigger proposal to NIH or maybe USDA as well. So we're still ironing out the details of this intervention. The study is going to start next year, the human part of that study. But yeah, so this is my research group, and they've really been, at least these 2 are joining us soon, but they've been instrumental in collecting the data for this. Onyedi Kenitai worked on the personalized Mediterranean intervention study. John and Shaheem are working on the exercise resistance training intervention. And then I have been an incoming student, Elahe, who actually just got awarded the Kapner assistantship, and then Eleanor as well. And they're going to be working on the Almond project and some of the other projects that we've got going on. So really, they've really been really instrumental in collecting the data for these projects. And then I'd like to thank our funding sources. The personalized intervention project is funded by the NIH, and the upcoming project is funded by the Almond project by the Almond Board of California. And then, of course, the donor award from the state, and very thankful for that. And we'll be working on the project based on that. And then all my collaborators, Rudy and Karina for the UC Merced project, and then Vicky for the Almond project, Jordan with the project, and Aaron, who's helping with the Microbial analyses. And this is my contact information. So if you'd like to get in touch with me, feel free to. I love to chat with people. But thank you very much for listening to me talk.
Jaapna, thank you for presenting your interesting research. Next, we are going to have Q&A session. So for the audience, if you have any questions, you can post in the Q&A session. I have a question for you, Jaapna.
Yeah.
I'm a food scientist, so from my point of view, I'm interested to know about those almonds, because there are different types of almonds. There are roasted almonds, branched almonds, salted almonds. So what type of specific almonds that you used in your research, and how much the volume that you gave to participants in the study?
So we've done different types. So I think during my PhD work, which is also on almonds, we did roasted almonds. And these are all provided by the Almond Board of California. Their farmers and the growers provide us the almonds. The one that we used for my postdoc work were not roasted, but they were whole and raw almonds. And we provided participants with about 2 ounces of that to eat every day. In some studies, we've done maybe like 15% of the energy intake if you really, really had to control the energy intake. But usually I think, the American Heart Association also recommends about 1.5 ounces of nuts is a good amount to consume for optimal health.
Great,
but I do sensory work on the side, nowhere to the extent that you do, but we track the palatability of the study foods and their participants' liking over time as well. And it's very interesting. You see some participants start enjoying almonds more over time, whereas some, you see the palatability decline a lot. They'll still eat it, but some of them don't want to see almonds again after 8 to 12 weeks of consuming it.
Okay, thank you. Another question that's posted by the audience is, how do other nuts compare to almonds?
Yeah, good. I used to have a slide for that in some of my other talks. But most nuts are similar in terms of nutrient composition. So there are more similarities than dissimilarities, I'd say. But the biggest is in terms of fat composition. So, for example, almonds are high in monounsaturated fats, and walnuts are higher in polyunsaturated fats. Now, both of these are the good fats. And nuts in general are low in saturated fats. So the studies show, especially in terms of immune health, a lot of these studies have been done with polyunsaturated fatty acids. If you go back a few decades, you'll see PUFAs and immune health, a lot of work in that area. And then walnuts are high in PUFAs. You'll see some studies based on that, but hardly or very limited studies looking at effects of almonds on immune health. So almonds and walnuts, I'd say, are the most popular nuts, or they've been the most widely studied in interventions. Not to say that, other nuts like pistachios or Brazil nuts, for example, don't have beneficial effects, it's just that they haven't been as widely studied.
Very good. Any more questions from the audience? You can post your questions, you can post in the Q&A session. Well, if no more questions from the audience, we are going to end today's seminar webinar. Thank you all for coming. And our next webinar will be displayed on a slide by the host, Rhonda. So it will be shown on the screen.
Yes, unfortunately, I don't have the information. I can tell you that it will be Rob Myers on April 21st at 3:30, Center for Regenerative Ag. That will be Kelly Wilson and Rob Myers. So I'll send that out in the Kafner info soon. Thank you, everybody.
Thank you, everyone.
All right, bye-bye.
Bye-bye.